simple blood test measuring 34 circular RNAs identified people at risk for Alzheimer's disease years before symptoms developed. In a study published in Nature Medicine, researchers analyzed samples from more than 3,500 individuals across multiple cohorts and found that this circular RNA signature outperformed the current blood biomarker p-tau 217 for identifying Alzheimer's disease. When the two tests were combined, diagnostic accuracy increased even further, achieving an area under the curve of approximately 0.98. To understand why this matters, consider what p-tau 217 measures. Tau is a protein that stabilizes the internal skeleton of neurons. In Alzheimer's disease, tau becomes abnormally phosphorylated, detaches from the cell's microtubules, and eventually forms the neurofibrillary tangles that are a hallmark of the disease. Elevated p-tau 217 is an excellent biomarker, but it reflects a pathological process that is already underway. Circular RNAs may reveal an even earlier stage of disease. Unlike conventional messenger RNA, they are edited into stable loops through a process called back splicing, making them remarkably resistant to degradation. Most are not translated into proteins. Instead, they help regulate gene expression by interacting with microRNAs and other components of the cell's genetic control system. Rather than measuring a single downstream protein, the researchers identified a 34-molecule regulatory fingerprint that appears to change before extensive tau pathology develops. Think of p-tau 217 as evidence that structural damage has begun inside a building. Circular RNAs may reveal that the blueprint and the way the construction crew is following it has already changed long before cracks appear in the walls. If validated in future clinical studies, circular RNA profiling could shift Alzheimer's diagnosis from detecting established neurodegeneration to recognizing the earliest changes in the brain's regulatory program, when intervention may have the greatest opportunity to preserve cognitive function.